Hello friends, welcome to Sam Biology class. Today we shall discuss about sex determination in humans. Humans like Drosophila have XXXY sex determination, but in humans there is a gene on Y chromosome known as SRY gene that determines maleness. Humans, both male and female, possess 22 pairs of non-sex chromosomes or autosomes. Both males and females possess 22 pairs of autosomes or non-sex chromosomes. But regarding sex chromosomes, there is difference between them. Males possess one X and one Y chromosome, but female cells have a pair of X chromosome. Because of the presence of two different types of sex chromosomes, 50% of the gametes produced by male parents will have one X chromosome and remaining 50% will possess one Y chromosome. Because of this, males are heterogametic. Producing two types of gametes. But in case of female, due to the presence of only one type of sex chromosome, all the gametes formed by the female parents will have one X chromosome. Thus, females are homogametic. And in human being, the inheritance of sex results in equal number of male and female offspring. This figure clarifies this. Male, female. Both male and female have 44 non-sex chromosomes or autosomes, and male have one X and one Y chromosome, whereas females have a pair of X chromosome. Due to the presence of two types of sex chromosomes, 50% of the gametes produced will have one X chromosome and remaining 50% will have one Y chromosome. In case of female parent, due to the presence of one type of sex chromosomes, all gametes formed will have one X chromosome. After fertilization, male and female offspring will be produced in equal numbers. So here these are the male gametes or sperm. And in case of human being, all female gametes that are produced will have one X chromosome. After fertilization, male and female offspring will be produced in equal number. So if the individual having a pair of X chromosome, it will be a female, and if it will have one X and one Y chromosome, it will be a male. So here also a female offspring will be produced. Here also a female offspring will produce, and in this pair, due to the fertilization of a female gamete having one X chromosome by a male gamete having one Y chromosome, a male individual will be produced. So in case of human being, inheritance of sex result in equal number of male and female offsprings. But sometimes, due to the presence of abnormal number of sex chromosomes, unusual phenotypes such as Turner syndrome, Klinefelter syndrome and X syndrome arise. The syndromes are Turner syndrome, Klinefelter syndrome and X syndrome. All these syndromes arise due to the presence of abnormal number of sex chromosomes. First one is Turner syndrome. Persons with Turner syndrome are abnormal females having only one X chromosome, whereas normal females have a pair of X chromosomes. Affected individuals have underdeveloped secondary sex characteristics. They have short height, low hairline, broad chest and normal inclusions. In most of the cases, they are sterile. Turner syndrome is seen in 1 of 3,000 female births. Second syndrome is Klinefelter syndrome, which is characterized by the presence of one or more Y chromosomes and multiple X chromosomes. Individuals with Klinefelter syndrome are abnormal males. They have small testes, mental retardation, and they have normal inclusions, and in most of the cases they are sterile. Klinefelter syndrome occurs with a frequency of 1 in 1,000 male births. In most of the cases, Klinefelter males have this combination 2X and 1 Y chromosome in their cells. But sometimes in few cases we can also get other combinations such as 3X chromosome and 1 Y chromosome, or 4X chromosome and 1 Y chromosome, or 2X and 2 Y chromosomes are also seen. Men with Klinefelter syndrome frequently have small testes, reduced facial and pubic hairs and normal inclusions. They are often taller than normal and are mostly sterile. The syndrome occurs with a frequency of 1 in 1,000 male births. Next one is X syndrome, which is characterized by the presence of 3X chromosomes. These individuals are known as X female, which are tall and thin. Many of them menstruate regularly and are fertile. A few of them are sterile. The syndrome occurs with a frequency of 1 in 1,000 female births. The incidence of mental retardation among X female is greater than that of general population. Individuals with 4 or 5 X chromosomes are considerably rare. They have a normal female anatomy and are mentally retarded. They are mostly sterile and have a number of physical problems. Individuals with 3 or more than 3 X chromosomes are known as poly-X female, Poly-X female. The severity of mental retardation among these individuals increased with an increase in the number of X chromosomes. Functions of sex chromosomes. Number 1. The X chromosome contains genetic information essential for both sex. At least one copy of X chromosome is required for the human development. Number 2. The male determining gene is located on Y chromosome. This gene is known as SRYgene, which was discovered in 1990. A single copy of Y chromosome, even in the presence of several X chromosomes, produce a male phenotype. Number 3. Absence of Y chromosome produce a female phenotype. Number 4. Genes affecting fertility are located on X and Y chromosome. A female usually requires at least 2 copies of X chromosomes to be fertile. Number 5. Additional copies of X chromosomes may upset the normal development in male and female, producing physical and mental problems that increase as the number of additional copies of X chromosomes increases. Next we shall discuss about the male determining gene in humans. This gene is known as sex determining region Y gene or SRY gene, which is located on the short arm of Y chromosome. This gene is located on the short arm of Y chromosome. Early during development, all humans possess undifferentiated gonads and both male and female reproductive ducts. About 6 weeks after fertilization, a gene on the Y chromosome becomes active. This gene is the SRY gene. The activated SRY gene causes the development of undifferentiated gonad into testis, cause the development of undifferentiated gonads into testis that begin to secrete testosterone and Mullerian inhibiting substance. One is testosterone and the other is Mullerian inhibiting substance. Mullerian inhibiting substance. Testosterone induces the development of male characteristics. Whereas Mullerian inhibiting substance cause the degeneration of female reproductive duct. Absence of SRY gene causes the differentiation of neutral gonads into ovaries, and as a result, female features appear. SRY gene encodes a protein called a transcription factor. SRY gene encodes a protein called a transcription factor that binds to DNA and stimulates the transcription of several other genes that promote the differentiation of testes. Although SRY gene is the primary determinant, of maleness in humans, other genes such as X-linked genes, Y-linked genes and still other autosomal genes also have roles in fertility and in the development of sex differences. So friends, this is all about sex determination in humans. For more updates, subscribe my YouTube channel, Some Biology Class.